So I think we're going to get going so that when inevitably the technical difficulties arise, we have an extra minute. All right, so thank you all for joining us today. We have an exciting session that's titled Rescuing CFTR Premature Truncation Alleles, Basic Biology and Novel Therapeutic Strategies. So co-chairing with me today is Jay Chen here from University of Alabama, Birmingham, and I'm John Lewick from University of Rochester. Here are our disclosures. Okay, so what are we talking about today? So genetic missteps that cause loss of CFTR protein and its function, and also reduction in the CFTR transcript abundance in area epithelia. So this arises in many ways. Today we'll be talking about duplications, inserts, and deletions. And just to kind of go over the basic genetic missteps that the therapeutics will be targeting. So these duplications, inserts, and deletions can be big and small. And the focus of these are the ones that put translation out of frame. And so I have listed a couple examples here. On top are duplications and inserts. Here you have a duplication of an AT that puts translation out of frame. So you have a premature termination, and then you have nonsense-mediated decay of the transcript. But you can also have duplications of entire exons and introns, and I'm showing here a rather large duplication of exon 26, intron 26, and exon 27. Oop. That's sensitive for the speakers for doing the trackpad. Okay. All right. Well, then this is weird. Okay. So, and then on the bottom we have deletions, and deletions can arise again through very large deletions and very small deletions. So, in two nucleotides, one nucleotide, and then obviously you can have deletions of entire swaths of the genome, 78,000 base pairs. You have nonsense and splice mutations. On top, I've listed some common nonsense mutations. This arises from one nucleotide difference from a coding codon to one of the three stop codons, TAG, TGA, and TAA. In an RNA, that's UGA, UAG, and UAA. And then on the bottom are splice mutations, and these are really interesting. They're not digital. I consider these analog, where you have a change in, listed on the bottom, donor sites, splice enhancers in both exons and introns, and also you can have silencers, so exon silencing mutations. And this is where now you have alternative splicing of the mRNA, which then again puts translation out of frame. You have truncated protein and loss of the transcript abundance. So now all of these have a common theme, is that you have loss of protein. Now the known therapeutic Trikafta has no target, right? Because you don't have the protein to act on. So now we need to come up with different therapies that will target either the genome or RNA. In many cases, likely a combinatorial approach. So the overview and topics provide an overview of basic electrophysiology, cellular biology, and protein chemistry related to understanding CFTR biology and development of technologies for better treatment of CFTR that are not missense mutations or delta F508. And then we're going to cover various topics that include nucleotide-based therapeutics, genome editing, viral and non-viral strategies to have a durable, efficient, safe rescue of CFTR function. And we'll also touch a little bit on delivery strategies. So we had a embarrassment of riches for abstracts to pick from. And so we've whittled it down to the 5 that you'll hear today. And we wanted to have a breadth of technologies, kind of different approaches for all of these different genetic missteps that result in CF. And so here, starting off, we're going to have Lucia Santos. Would you like to come up? And so the way that the questions will go, speakers have 15 minutes, 8 minutes of questions. And so hold your questions to the end. And if we end early, maybe we can have kind of an open question session at the end. All right, so now this is the difficult bit. Yeah. Let it rip.
Yeah. Good morning, everyone. Thanks for the opportunity to present today. I have nothing.
Yeah, advance using this. Or you can use that.
It's important.
Oh, boy.
Well, maybe I can use this.
There you go.
Okay. I have nothing to disclose. So, as all of you know, in the past decade, 4 different CFTR modulators have been approved, from which 90% of the patients can benefit. And even though these have been a major breakthrough for CF therapeutics, there are still 10% of the people with CF that do not benefit from any targeted therapy. Patients carrying W1282X mutation belong to this 10%, and this mutation is caused by a G to A modification in exon 23 of CFTR that introduces a premature stop codon that leads to mRNA degradation by nonsense-mediated decay, and consequently, no protein or very little protein is produced. Patients could potentially benefit from read-through drugs or NMD inhibitors, but none of these have been approved yet. So genetic-based therapies can potentially provide a one-time cure, avoiding a lifelong treatment. Our lab have been using different genome editing strategies to correct different CFTR mutations, but for the purpose of this presentation today, I'm going to focus on adenine-based editing that can potentially correct 28% of all CF-causing mutations. And how does adenine-based editing works? So we have the guide RNA that targets the Cas9 nickase to the target site that nicks only one strand, one of the DNA strands. And then the adenine deaminase converts adenines into guanines. And the adenine deaminase works in a specific editing window of nucleotide 4 to 7 in the spacer sequence, counting the PAM as nucleotide 21 to 23. And in this example here, we have the guide RNA used to target W1282X mutation that puts the A that causes the mutation in position A6, but there's another A in position A7. And previous studies have used adenine-based editing to correct W1282X mutation in primary human and HEX cells. And what these studies show is that there is a high editing efficiency of the A that causes W1282X, the A6, but there's also editing of the A7. And to this we call the bystander editing. We develop an additional cell line carrying W1282X on the BCI cells background, and we did this by CRISPR-Cas9. We isolated clones from the pool of edited cells and confirmed the genotype by Sanger sequencing. And then we characterize these cells. We assess the levels of CFTR mRNA and confirm a decrease in the mRNA levels in our W1282X homozygous clone. And there's no fully glycosylated protein being produced. These BCI cells, they have the capacity to differentiate in all known cell types of the lung epithelia. And we assess the levels of cell type-specific markers, such as keratin 5 for basals, FOXJ1 for ciliated cells, MUC5AC for goblet, and FOXI1 for the ionocytes. And we confirm that over time that our W1282X cell line can differentiate in all known epithelial cells. We then transfect our novel cell model with the SPRY ABE 7.10 to which we called the intact ABE. And we observed what had already been observed previously. So there's editing of the A6, the target A, but there's also alleles where the A7 is edited, alleles where both A's are editing, and that adds up to 12% of total editing. So we demonstrated that adenine-based editing can efficiently correct W1282X with minimal indels, but there's significant bystander effects. So an improved adenine-based editing tool, or ABE, is needed. In this paper from 2015, the authors split the Cas9 nuclease into 2 halves. the N-terminal containing an FRB domain, and the C-terminal containing an FKPB domain. And only in the presence of rapamycin, these two halves would assemble and then edit the DNA. And by splitting the Cas9 nuclease, the authors observed that minimized the duration of the nuclease activity, reducing the off-target effects. So we wonder if by splitting the Cas9 nickase in the ABE, if that would reduce the bystander effect. So we came up with two different constructs. We have the U6 promoter to express the guide RNA, and then ubiquitous promoter to express our split ABE. So we have the C-terminus with the FKPB and the N-terminus with ABE and the FRB domain. And the difference between these two constructs is that the one at the bottom carries nuclear exportation signal. So we then evaluated our split ABE and compared with the intact ABE. And in the absence of rapamycin, we observed that there's still background editing that comes down when we have our nuclear exportation signal in our construct. In the presence of rapamycin, these levels of editing go up, but more importantly is that minimal bystander effects are observed. So in summary, we developed a split ABE that can successfully correct W1282X, reducing the bystander effect. We are evaluating the capacity of this split ABE to correct other nonsense and splicing mutations. The split ABE, the regulation of the split ABE by a small drug gives us temporal control of the editing. And we are now developing a system to for cell type specific expression of the split ABE that will allow us to have spatial control of the editing. And that will allow us to achieve our long-term goal, which is to understand which cell types and how many of them we need to correct to restore CFTR function to therapeutic levels. So I would like to acknowledge people at Dr. Carlos Farinha lab in Lisbon and Dr. Patrick Harrison's lab in Cork, and Cystic Fibrosis Foundation for the funding.
Plenty of
I still have 7.
You still have 7. Questions? Oh. Questions?
Yeah, nice presentation. Garrett Cutting, Johns Hopkins. So why does splitting the ABE decrease bystander effect?
That's what we are still evaluating. We don't know yet how the bystanders are reduced. It might has something to do with the structure of the protein when it assembles. We have some preliminary data showing that I didn't present here today because it's too preliminary. But it seems that the ABE edits preferentially the first A in the editing window, and that's the case here. And maybe it edits the first A and then detaches the DNA, and then when it comes, the first one is already edited and edits the second one, but not as efficiently.
Okay. Thank you.
Thanks for the talk. This is Yuan from B. Riley. Just curious, have you tried to lower the expression of your ABE to see how that will impact the overall correction ratio? For example, Y is at 5%. Can you increase it to higher or lower? And in that case, if it's lower, will the second ABE or bystander effect be reduced?
We have some experiments planned to do a dose response, increasing the levels of rapamycin to see if by increasing the levels of rapamycin, we have more protein expression, and then increase the levels of editing, and to see if that will increase the levels of bystander or if we keep them the same.
All right, thank you.
Oh boy.
Oops.
Push escape. Well, interesting. Okay. Well, don't you worry about that. Go ahead and answer the question.
Hi, Lucia. Thanks so much for a nice talk. I had a question. So in this case, your edit of interest was your A6 and your bystander was A7. And I think you kind of answered this in response to Gary's question, but have you looked to see whether or not that's a consistent effect? Like if you have other targets that had an A6 and A7, but maybe A7 was the one you wanted to edit. So I'm just kind of wondering, like here we're defining it as a bystander, but could you comment more on like the editing window and if you've looked at other targets?
We did, not on CFTR, but none of those are A7. So we have a target where we have A5 and it's A5, it's the first A, and that's the one that it's edited most efficiently. And then we have another A at A7, and the levels are much lower in the case. Okay. We haven't tested the target where the A7, it's the first A in the editing window.
Okay, thanks.
I think we'll have time. I have another question. So did you have to play with the expression of the front and back half of Cas9 to get this to work more efficiently, or are you just hoping that, equal expression is going to give you optimal editing? So you have to put this in two pieces, and you use a P2A.
Yeah.
Have you done it on two constructs and expressed more of one and the less of the other? Okay.
No.
Any other questions? All right, let's thank Lucia for a great talk.
Thanks.
Let's also thank her for finishing early because we have technical issues that we need to resolve. Thank you for that. All right, we're back. So next up we have Javier Campos-Gomez, and we're ready to go. You ready? All right.
Okay. Good morning, everybody, and thank you for allowing me to present this work here. And first, I am disclosing that Arcturus Therapeutics sponsored this study. And to start with some background information, lunar CFTR is a novel proprietary aerosolized CFTR replacement therapy aimed to treat CF patients carrying mutations that do not benefit from available CFTR modulator drugs. Lunar CFTR consists of a codon-optimized CFTR mRNA encapsulated in lunar lipid nanoparticle technology for efficient delivery into the airway epithelial cells. And in this study, we evaluated whether lunar CFTR was able to restore CFTR expression and function in terminally differentiated human bronchoepithelial cells derived from CF patients carrying different CFTR mutations. So basically, we will evaluate how efficient is the lunar LNP technology at delivering mRNA to CF bronchoepithelial cells, and how good is the optimized CFTR mRNA delivered at expressing the protein and restoring its function. So to evaluate the lunar technology, we use human bronchoepithelial cells from CF and wild-type donors as control of normal CFTR function. And the cells were fully differentiated at the air-liquid interface using vertex medium in the basolateral side of the cells. And for transfection with lunar nanoparticles, the differentiated HB cells were washed briefly to remove the excess of mucus and incubated with the desired amount of lipid nanoparticles for 6 hours. And lunar TD tomato and CFTR were always used in parallel, in different transwell, but in parallel, to be able to assess the transfection efficiency by quantifying the fluorescence. In this case, we quantified the fluorescent area in the filters using an IMASJ algorithm. And finally, we measured the CFTR function by measuring the short circuit currents after 24 hours post-transfection, either in the presence or the absence of a chloride secretory gradient during Usen chamber measurements. And I would like to remark here that cell monolayers were always quality controlled during electrophysiology studies, and filters showing lower than 100 ohms per square centimeter baseline transepithelial resistance were discarded from the CFTR function studies. So initially we used HBE cells from 3 different CF donor carrying the delta F mutation, and from 3 wild-type donors to assess the transfection efficiency and CFTR function. Here you can see a representative microscopy image of an entire transwell filter of CF cells transfected with the lunar TDT tomato compared to a mock.
You can move the
What is
cursor.
that?
Move the cursor to the other screen. There you go.
Compared with a mock transfection. And in general, despite the donor-to-donor variability, we showed that the fluorescence was very similar in CF compared to wild-type cells at around 34% transfection efficiency. We took away our concern of potentially less efficient transfection in CF cells due to a thicker mucus barrier. Remember here that I briefly washed the cells before transfecting, but I have tried few filters without washing, and the transfection is kind of similar. So subsequently, we evaluated the dose-response relationship of the lunar CFTR to guide dosing for future experiments, and we tested lunar CFTR using 2.5, 5, 10, and 20 micrograms of the mRNA in CF cells, and as you can appreciate, incremental doses of the mRNA also produce proportional increments in the Forskolling response. And also Western blotting specifically for CFTR protein showed that CFTR expression shows CFTR expression as a function of dose, and that cells expressing in fact higher protein compared to the wild type at doses higher than 5 micrograms. So we use the 20 microgram dose for most of the experiments I'm going to show you today. So in the past, we have tested other lipid nanoparticles that failed to show good expression and restoration of the CFTR function. Thus, to maximize the effect of lunar CFTR, we initially tested the transfected cells in the presence of a chloride secretory gradient during Usen measurements. And in this slide, you can see representative current tracings obtained in the presence of a chloride gradient, and the TD tomato transfected cells show the typical characteristic of the CF cells. I mean the typical tracing of CF cell with low response to Forskolling and the modulator BX770. However, well, compared to the wild type transfected with the same TD tomato, however, the CF cell transfected with lunar CFTR showed the typical tracing of wild type cells, but they show a higher BX770 response, and they didn't show the FSK peaks that the wild type usually show. So after extracting the Forskolling induced current values from the tracings, you can see that the CF cells transfected with lunar CFTR show significantly higher Forskolling response compared to the TD tomato transfected cells, and similar to the wild type transfected cells. And in the summary graph to the right, you can see also the contribution of the BX770 to the total current, and that the currents were specifically inhibited by the CFTR inhibitor 172, indicating that indeed the currents were CFTR specific and not leaky current due to alteration of the transepithelial resistance. So we next evaluated the effect of lunar CFTR in accent of chloride gradient, which is a more realistic clinical scenario. And in this case, the tracing also showed that CF HBE cells transfected with CFTR mRNA exhibit significantly higher chloride current compared to the CF cells transfected with lunar TD tomato. Yeah, here. And although the amplitude of the response was lower than that obtained with the presence of the chloride gradient. So again, here we saw a more pronounced BX770 response and a less pronounced FSK peaks compared to the wild type cells. So in average, the FSK stimulated response of CF cells treated with lunar CFTR in the axon of a chloride gradient was around 60% of the wild type response. However, lunar CFTR induced chloride current comparable to those of the ETI modulator. And in the summaries graph, again, you can see the contribution of BX770, and that also, in this case, the current was specifically inhibited by the INH 172. So we think that the reduced CFTR function in the axon of a chloride gradient, despite a robust CFTR expression measured by Western blot, may indicate transfection of airway cell subpopulation not specialized in secretion, which have minimal impact on the chloride ions transport in the axon of a chloride gradient. But when you impose a chloride gradient, these cells are able to somehow contribute to the current. Alternatively, other explanation could be that the overexpression of CFTR may cause mislocalization of the protein to the basolateral membrane, which partially cancel out the chloride current on the into the apical direction, or that a significant proportion of the protein is misfolded, although we have seen in the Western blot that the majority of the protein is in the C band, in the correct folded protein. So subsequently, we evaluated the time course effect of lunar CFTR to design a repeated dose experiment using lower amounts of the CFTR mRNA. And then using the 20 microgram dose for transfection, we determined that lunar CFTR has a half-life of around 3 days in CF cells. And using this result, we tested lunar CFTR at 3 and 5 microgram doses every 3 days for a total of 5 times. And as you can see in the graph to the right, the 5 microgram dose induced chloride currents comparable to a single dose of 20 micrograms measured in the axon of a chloride gradient, and to the currents induced by ETI in the same CFHB cells. So in all the previous experiments, we use CFHB cells carrying the delta F mutation to be able to compare to lunar CFTR response in the presence of ETI modulators. Thus, we further evaluated the effect of lunar CFTR in CF-HBE cells carrying null mutations, which are unresponsive to CFTR modulators. And in this case, again, we observed that lunar CF formulation have a significantly higher FSK response compared to the TDT transfected cells, and approximating those responses of the wild type cells. So to summarize, we have extracted as main conclusion from our study that lunar CFTR successfully transfected and expressed a functional CFTR protein in delta F and null CFTR HBE cells in a dose-dependent manner, that in the presence of a chloride gradient, lunar CFTR treatment restored chloride currents in CF cells to levels approximating those of the wild-type cells. In the absence of a chloride gradient, lunar CFTR induced chloride current at 60% of wild-type current level, but was comparable to those of the ETI, the clinically approved CFTR modulator for CF patients. And that repeating dosing of 5 micrograms of lunar CFTR administered at a half-life time interval achieved chloride current comparable to a single 20 microgram dose and comparable to ETI-induced currents, indicating a beneficial effect of dose accumulation. And to conclude with our current and future studies, lunar CFTR is currently being tested in phase 1 clinical trials in healthy volunteers and adults with CF, and the identification of cell types transfected and CFTR localization are in progress to determine why, despite the higher CFTR expression in CF transfected cells, the currents are still lower compared to the wild type. And to conclude, I would like to acknowledge Marina Masur, that produced all the HBE cells used in this work, and Lipin Tan for her technical assistance, both from UAB, and the RNA science and CF formulation team from Arturus, and the Cystic Fibrosis Foundation for funding assistance. And with this, if you have any question, I will be glad to.
Thanks for the talk. Just a couple of questions. First, when you have different doses and the transfection efficiency was at about 30–40%, when you increase the dose, have you observed increased transfection efficiency there?
You mean with the TD tomato protein?
Yeah.
Yes. I always did the experiments in parallel, and I see also a transfection increase in the fluorescent. It's not shown here, but yes.
So why is stopped at 30%? Have you observed, for example, 60% of transfection?
60%? No. Well, in some filters, yes. But as an average, it's a maximum of 40% usually.
Got it.
But some filters are outliers always, and you have more and less.
Got it. And then a follow-up question is, why is the CFTR?
Although I suspect that I am underestimating the transfection efficiency, because I only quantify the area of red. So sometimes when you lost some low expression because IMAGE is not able to see lower expressing cells, that they might be expressing the red fluorescent protein, but you don't see in IMAGE because the contrast.
Got it. And so in that case, if you are not using fluorescence, if you use the protein staining of the CFTR protein, what's the expression level you observe there using mono?
Well, I have not done a staining for that, but the Western blot surely indicate that. So you see in the Western blot that I presented that the expression also increased proportionally to the dose that you are using. And in fact, it's a very high dose compared to the wild type even.
Got it. And one last question. For the CFTR expression, why the half-life is only about 3 days? What happened there? Is it because the CFTR protein is not stable?
Well, I think that's the normal. Every protein has a half-life, and mRNA has a half-life. I think this is a combination of the half-life of the mRNA and the protein. I also did it in parallel with the TD tomato, and TD tomato have a longer half-life than CFTR.
Got it. And then for your platform, I think you guys are using the self-amplifying mRNA. Does that supposed to have a longer half-life for the mRNA and hence the CFTR expression?
Sorry.
The self-amplifying mRNA in your Luna platform.
Yeah, this CFTR function is not based on cell.
Got it.
No.
Got it. Thank you.
Hey, thank you very much for the presentation. This is Lin from Prime Medicine. I wonder, like, before clinical trial, are there any in vivo data for this in mouse or other animal models?
Yes, but not from our group. The Iowa University is working with these lipid nanoparticles in ferrets, and they have seen also promising results.
Would you like to introduce more about, like, in ferret, which part or what cell types are these LNP successfully targeting?
I don't know.
Okay, thank you.
I'm not doing that work. I can tell you about the cell type that might be transfecting in.
I just have one.
Well, in fact, we are studying that now.
All right. I just have one question. So the mucus layer is an important issue, right? Barrier. Mm-hmm. And you said you washed that mucus layer off.
Yes.
Have you tried the experiments with your LNPs, leaving the mucus layer on there to see?
Yeah, I mentioned that I have done that with few. I didn't present the result because I have done ultimately lately, and it's not here. And with few filters, I would like to repeat more. But apparently, it's very similar. But this is Vertex medium, and I have noticed that Vertex secretes less mucus than pneumococcal, for example. In pneumococcal, I see worse response, but still is there, even with pneumococcal.
All right, I did have a second question. So are these pegylated?
Sorry?
Are they pegylated? Did you
pegylated?
Peg?
The nanoparticles?
Yeah.
I don't know the
Secret formulation?
because this is proprietary. I don't know.
Okay.
I'm a blind user of the lipid nanoparticles, so I don't know.
They are, okay?
My question's about the transcript is codon optimized.
Yes.
So increased translation rates, you might decrease, co-translational folding efficiency.
Yeah.
The defects you're getting in the CFTR protein, did you try like a non-codon optimized lunar transcript to see if that goes away?
No, I haven't tried, but if you see in the Western blot, this is an example of Western blots, the majority of the protein is in the C band, although you have a lot of protein that is misfolded. That I mentioned that could be one of the explanations that the current in the absence of the chloride gradient, even with this very high expression, is lower than the wild type. But I tend to favor the hypothesis that we are transfecting many cell types that are not specialized in secretion. And so when you force a gradient, they contribute to the current. But when you don't put a gradient, those cells are contributing very little to the total current, and only the specialized cells, the secretory cells, are really producing the majority of the current. But that's because another hypothesis is that the excess of protein, part of them is going to the basolateral membrane, and they produce a current in the other direction that cancel the proper directional current. But I just last week have done some immunostaining, and I see well localized in at least the majority. I see a lot of green in the apical membrane and not in the basolateral. So I tend to favor more the hypothesis that subpopulation of cells not specialized in secretion are contributing to.
Okay, thank you.
Time for one more question. Quick one.
Thank you so much. So my name is Clemente Brito. If you induce a pro-inflammatory state on that epithelial layer, would that affect the correction effect by the lunar CFTR? For example, if you gave IL-8, IL-6, TNF-alpha, the things that we find in the airway environment all the time?
I guess that yes, but I never done that. But I guess that probably yes.
Yes, it would increase correction, or yes, it would affect correction in an unknown direction?
I don't know. I don't know.
It'll be different.
Thank you very much.
That could be speculating from my. I think could be either, but maybe I will tend to think that it will affect. It will downregulate.
Thank you so much.
Actually, I do have one question. So, if you increase the amount of delivering amount of your lunar in mRNA, and are you just getting more into the same cells that would have received the mRNA, or are you delivering it to more cells? And if you're delivering it to the same cells, just getting more mRNA into those same cells, are you saying that there's cells that are refractory to delivery?
You mean when I do the repeated dosing?
So you go up to 20 micrograms of mRNA when you deliver. You get an increase in response. Is that because you just get more mRNA into any given cell, or are you getting it into more cells? And if you're not getting it into more cells, are you saying that some cells are refractory to delivery?
No, I think the more you use, more cells expressed, because that's what happened with CD tomato.
Yep.
A higher amount of cells will express that, but also you see many red intense cells, meaning that they
Take up more.
take up more. But if you do repeated doses, what I have seen is that with those repetitions, you see more homogeneity in the red on the well compared with the previous doses.
You're getting different cells.
Yes, you
Gotcha.
guess that different cells are getting it there.
All right. Well, thank you very much.
Okay, our next speaker is Alice Eastman. She's from Johns Hopkins School of Medicine. Her topic is functional and RNA level recovery of CFTR nonsense variants with NMD inhibition, read-through, and ETI. Alice, please.
Thank you so much. Hello, everyone. My name is Alice Eastman. I'm a sixth-year graduate student in the Human Genetics and Genomics Department at Johns Hopkins School of Medicine. I work with Gary Cutting and Dr. Neeraj Sharma, and I'd like to thank our organizers of this year's conference and our moderators for giving me the opportunity to speak today. Today, I specifically want to tell you about our work entitled Functional and RNA Level Recovery of CFTR Nonsense Variants with NMD Inhibition, Read-Through, and ETI.
Just go ahead first. You got it?
Yeah. I have no relationships to disclose for this presentation. So I'd like to start with an idea about the motivation behind this project. According to the CFF, around 7% of people with CF have a combination of two nonsense and rare mutations that do not produce CFTR protein. Because these individuals do not produce CFTR protein, they cannot benefit from CFTR modulators. They will need different therapies to fix the underlying cause of their disease. As a brief reminder of what nonsense variants are, we can start with the classic central dogma of biology. DNA is transcribed into RNA translated into protein. A nonsense variant is one which produces a premature termination codon, or PTC, in the DNA, which is transcribed into the RNA. Any protein formed from that mRNA will be truncated, but frequently mRNA with a PTC is targeted for destruction by the cell in the process of nonsense-mediated decay, or NMD. I want to point out that there are compounds we can use to allow for read-through of premature termination codons, which can lead to full-length protein, but this is only effective when there is stable mRNA. Here, I'm only trying to emphasize that nonsense variants in CFTR have heterogeneous outcomes depending on their location in the gene and surrounding sequence context. Here, I'm showing where a selection of nonsense variants would fall in the CFTR protein, and I'm noting what kinds of effects they actually have. As you can see, some of these variants result in efficient NMD and very low mRNA levels, where others evade NMD. Additionally, if protein is made, the quantity and quality of the resulting protein varies based on the context of the initial variant. This heterogeneity implies that we may need different types of treatment options depending on genotype. Today, I will mostly be showing you results from the W1282X variant, which leads to low mRNA levels, but partially functional truncated protein. To systematically evaluate CFTR nonsense variants to inform therapy, we start by asking a few questions. First, we want to know which CFTR nonsense variants make stable mRNA, which make mature truncated protein, what is the level of function of any CFTR produced, does that protein respond to modulators, and finally, can NMD inhibition, NMDI, augment CFTR modulator response? To answer these questions, we systematically analyze nonsense variants and splicing variants in CFTR using an expression mini-gene or EMG system. In this system, either full-length or abridged introns are inserted into CFTR cDNA and plasmid. Using introns allows for splicing, which is necessary to recapitulate NMD or missplicing seen in people with these variants. Variants created in EMGs are shown here on this illustration. And here I'm showing nonsense and frameshift variants in red and purple, intronic variants in blue, and misspliced variants predicted to cause, missense variants predicted to cause missplicing in green. For example, W1282X is an exon 23. So to study this variant, we use an EMG with abridged introns 21 through 24 along with the variant. With these plasmids containing EMGs with selected nonsense variants, such as the W1282X EMG construct on the left, we transfect flippin stable cells. Specifically, we use HEC293 cells to study protein levels with Western blot, and CFBE cells to study RNA by qRT-PCR and bulk RNA sequencing, as well as function by short circuit. In addition to these stable constructs, we have cultured primary cells from people who have these variants. Here we use human nasal epithelial or H&E cell culture, which is a well-established technique that has been used widely for CF and other research. For this technique, we acquire nasal airway cells by brushing, suspend them in media. Then conditional reprogramming is achieved by plating the nasal cells on a feeder layer of irradiated 3T3 mouse fibroblasts and treating with ROCK inhibitor. Cells are expanded, then differentiated at air-liquid interface culture. With differentiated cell monolayers, we are able to perform functional analysis in Usting chambers, which I'll describe in a little bit more detail soon. To study nonsense variants in primary cells, we recruited 41 individuals in 3 categories. Individuals with CF with at least one nonsense variant, people without CF who are carriers for a nonsense variant in CFTR, and individuals without pathogenic CFTR variants. On the right, I'm showing the drugs we used in various combinations. So for NMD inhibition, we used antisense oligos, or ASOs, directed to elements of the NMD machinery. Specifically here, we use SMUG1, SMUG6, and UPF3B ASOs. For read-through, the data I'll show use either G418 or ELOX02. And finally, we used CFTR modulator combination, lexicaftor, tezacaftor, ivacaftor, also known as Trikafta, which I'll call ETI. So transitioning to the results, I'd like to start with CFTR RNA expression in our W1282X CFBE cells. So on the left here is CFTR RNA expression levels normalized to HPRT1 levels using qRT-PCR, which is a quantitative and sensitive measure for relative transcript abundance. The first 3 columns are treated with a scrambled ASO that's not directed to anything. And the next 4 are treated with SMUG1 antisense oligo. So here we can see that CFTR transcript is greatly increased in the SMUG1 antisense oligo treated cells compared to the scrambled cells. And then on the right, I'm showing that we can see that same pattern in bulk RNA-seq. And this is from RNA that's extracted from the same extracted RNA. To investigate protein quantity and maturity, we performed Western blots with HEC293 cells stably expressing our W1282X EMG construct. So for simplicity, I'll define C band as mature and B band as immature CFTR. In the untreated lane, we can see truncated mature protein, and recall that this does have some function. Treatment with Alexacaftor, Alexantiza alone or read-through agent ELOX-02 alone show little change. Then for NMDI, by treatment with SMUG6 ASO and SMUG6 ASO in combination with Alexatiza, there's an increase in truncated mature levels. And finally, when NMDI and read-through are combined, and when Alexatiza is added to that mix, we see that higher levels of protein are present, and we start to see full-length mature. Oh, it's on there. Full-length mature protein. So to measure CFTR functionality, we use short-circuit current measurement. To briefly go over this concept, we take filters that we grew the differentiated cells on. We mount them in Ussing chambers, which are equipped with electrodes to measure ion flux across the channels. Here we start with the same chloride concentration on either side. When we add an activator of CFTR, like forskolin and IBMX combination, we see an increase in current as chloride crosses through CFTR. Then when we add a CFTR-specific inhibitor, such as INH172, we see a drop in current, which we can use to quantitatively measure CFTR function. So here I want to show some short-circuit data from CFBEs containing the W1282X EMG. Baseline untreated function in blue. Do I need a laser pointer for this?
You can move your mouse to that. There you go.
Yeah. Baseline untreated function in blue is minimal. I'm going to go without it. But treatment with NMD inhibition by a SMUG1 ASO. Oh yeah, that might be better. Okay.
There you go.
Yeah. Okay. I'll use it when I need it. It'll be okay. Treatment with NMD inhibition by a SMUG1 ASO. Read through G418 and ETI each increase function, and then what I really want to show is that the combination of all three leads to the highest level of function. And then here on the right, I'm showing that we get the exact same sort of tracing when we use SMUG6 ASO instead of SMUG1. Finally, H&Es from human nasal epithelial cells from an individual homozygous for W1282X, we see the same result. So on the left, the initial current drop shown here from the addition of miloride is attributable to ENaC activity. Then on the right, I've zoomed into the CFTR portion of the show. Here we can see untreated in blue again, G4 and 8 read-through in green, ETI and cyan all have minimal function. Treatment with SMUG6 ASO and ETI in the dashed cyan have a little bit of something going on. And then again, the triple combination, we're really seeing more increase in function. And that's about 15% of wild type function that we're getting there. Here, I wanted to show some of the broader picture of these combinations of treatments, how they're influencing CFTR function in W1282X CFBs. And then before I conclude, I'll introduce a couple more variants for your consideration using this EMG system. So in this bar plot, I'm showing short circuit results for each treatment combination we performed on the CFBE cells. On the far left is untreated, followed by treatments with the individual compounds, then pairs, and then again, the final 4 bars show CFTR function for cells treated with NMDI, read-through, and ETI together, and those are showing the greatest level of function. I mentioned that nonsense variants in CFTR are heterogeneous. So when we try some of these same treatments on other nonsense variants, we get different results. So here I'm showing that S1196X and W1204X CFBEs on the same EMG background, the introns 21, abridged introns 21 through 24, show very similar results, but that R1162X has very little recovery from this treatment combination. So in conclusion, treatment with combination NMDI, read-through, and ETI can restore CFTR, RNA, protein, and function from nonsense variants, including W1282X, S1196X, and W1204X, but not R1162X. And I want to mention, of course, that variants like R1162X will require a different treatment approach, many of which we're talking about today. Thank you very much for listening.
Questions?
Hi, thanks for your talk. So basically, I was wondering about the NMD inhibition, which seems quite challenging in a clinical setting, right? No clinical trials going on right now. And I think one of the major concerns is safety and off-target effects, because it's obviously a normal cellular mechanism. So you did this bulk RNA sequencing on NMD-inhibited cells versus none. And I was wondering if you saw anything super interesting or toxic events or pathways that popped up.
And of course, that's why we were doing bulk, and I only got to show a very, very small fraction of the bulk that we did. So we saw very little toxic effects. So we chose, I have an extra slide for this guy. So here we have the setup. I was talking mostly about SMUG1 and SMUG6. So SMUG1 is involved in all NMD, right? But SMUG6 is really only involved in the endonuclease portion, which we know does degrade CFTR nonsense variants, but it's not required for all NMD. So that's one of our that's sort of our reasoning behind using some of these very targeted antisense oligos that are not required for all the NMD in the cell, because you're right. NMD is also a very important quantity control measure in addition to a surveillance mechanism.
Okay, cool. Well, if you didn't see pop up like major KEGH pathways or GO-term apoptosis related stuff, then that's quite good, right? Yeah. Nice.
I have a quick question. So in your SLIC kind of mini gene system, have you tried changing the expression level to see if, because you're using a pretty high expression of your transcript, if using a lower expression, medium, high, changes some of your outcomes? I was always kind of curious about that being, I mean, it's a very cool tool, and it's maybe the only way you can do as many things as you have, but does expression level affect your outcomes?
So we use a CMV promoter for all of these guys. I'm not sure if Niraj has tried other promoter. I don't think so. That's definitely something we could try. We measure levels of RNA expression with each of these variants that we're creating with QRT. That's an interesting idea to try for sure.
Only asking because CFTR is expressed so low, right?
Right.
John?
Hey, that's a great talk. I guess I was wondering, the environment with, translation and all the things, it's a very dynamic, and it's not necessarily just a clear stepwise fashion with NMD and read through. I guess I'm wondering if you could have a really high efficiency compound that would allow read through to occur, and you would get a lot of ribosomes on the mRNA, and you would get a lot of that translation. Do you think that an NMD inhibitor would be necessary with that, or would, because you're getting a lot of read through,
Right.
it would allow you to produce a lot of protein from there?
So I do think in general you'd want it. Again, some of these really don't go through much NMD. And if you have 80% of the transcript, then no, I think you could probably get by without the NMD. Other of these variants, you get 5% to 25% of the mRNA for CFTR, so some yes, some no is what I would say, and that is just going to depend on how efficient the NMD is for the specific variant.
Hey, a lot of work. Good talk. The size of the current in the CFBE cells was massive, like 50 to 100. In the primaries, it was much, much lower. Now,
Yes.
my understanding from your presentation is that the donors of the nasal cells are compound heterozygotes with the exon 1 allele. So what is the effect of the treatments on the other allele?
Right. So the ones I showed were homozygous for W1282X. But you're right. So many of the ones that we have from people with CF, the other allele is null. So we have R1162X with a null allele on the other side, an insertion. And we're also, sort of to get at that, we're also studying parents and sibling carriers. So we are really interested in how that other allele is affecting what's going on. For the difference in current that you were mentioning, we were comparing those to the wild type equivalents in their categories. So for the CFBEs, we were getting about close to 50% of functional recovery, and for the H&Es, we were getting about 15. But yeah, the scale difference is massive, and that's just due to the system.
Thank you.
That's it. Let's thank the speaker.
Yeah. Okay. Our next speaker is Dr. Ozeri-Galai.
Oh, you're a doctor. Not yet.
Not yet. From the University of Rochester Medical Center. The topic is one ACE tRNA sequence can functionally rescue the most prevalent CFTR nonsense mutations. Thank you.
Good to go?
Yeah, thank you so much for the introduction. So my name is Ozeri-Galai. I'm a grad student in John Luke's lab at the University of Rochester Medical Center. And today I'm going to talk about our recent work in the lab, which shows the promise of one ACE tRNA sequence for the most prevalent CFTR nonsense mutations. So here's the disclosure slide. And although the previous speaker did a good job of explaining what nonsense mutations are, I'll just do again. Nonsense mutations, or PTC mutations, are the cause of about 11% of all genetic disease due to single nucleotide mutation that convert an encoding codon into one of 3 stop codons. As a result, unlike normal translation that leads to production of normal functional protein, nonsense stop codon leads to production of truncated non-functional protein as well as nonsense-mediated mRNA decay, resulting in almost complete loss of gene expression and activity. So there are many different mutations in the CFTR gene have been identified that affects at the different stages of the CFTR processing pathways. And as many people in this room knows already, that there are 2 main CFTR modulators that's already approved for CF patient use, correctors and potentiators. However, because these modulators target malfunctioning protein, CF patients with nonsense mutations, which account for approximately 10% of CF cases, cannot be benefit from these modulators. And currently, there is no other treatment option available for these patient populations. So, as I mentioned, there are two main problems with nonsense mutation, and that can be targeted. So one is to somehow increase the amount of the full-length protein, and another one we could do is to somehow increase the abundance of target transcript by decreasing the NMD pathway activation. So currently there are different methods that are being explored, like genome editing, total gene replacement, or targeting NMD pathway or translation processes. The therapeutic approaches that our lab is interested in is the anticodon edited tRNA or ACE-tRNA. So tRNA is recognized by amino acid tRNA synthetase and charged with canonical amino acid. Then they participate in translation by interacting with its cognate codon. But what we wanted to do with ACE-tRNA is to interact with the disease-causing PTC and suppress nonsense stop codons. So we mutated the anticodon of the tRNA are still charged with the canonical amino acid, and then this ACE-tRNA now interact with disease-causing PTC instead of its cognate codon. So the translation process can be continued and make full-length functional protein. So in this data from our recent paper, we aim to insert natural amino acid at the PTC site. So we use we use the construct expressing both ACE-tRNAs and amnion green fluorescent protein, and transfected 16 HB cells with this construct and sorted the cells into amnion green negative and positive cell populations and measured the CFTR mRNA expression level. And the cells that expressed ACE-tRNA had significant improvement in the CFTR mRNA expression level. So glycine ACE-tRNAs for G542X, arginine ACE-tRNAs for R1162X, and leucine ACE-tRNAs for W1282X, but not with tryptophan ACE-tRNAs for W1282X cells. The reason we included the leucine ACE-tRNAs is because we really have bad tryptophan ACE-tRNAs. And second, it's been shown from Dr. David Bedwell's group that leucine at these positions can rescue CFTR function close to the wild type level. And lastly, we really had good leucine ACE-tRNA, so that's why we use this. So from this result, we wonder if this increase in CFTR mRNA level translate into increase in CFTR function. So we performed the PASCLAM experiments of cells transfected with the same construct. And the cells were pre-treated with Forskin and IBMX to maximally activate CFTR, and then added with the CFTR inhibitor 172 to inhibit channel activity. So the current changes in response to CFTR inhibitor is the measure of the CFTR function. And for the dotted line expressing empty plasmid, we did not see any functional CFTR. But ACE-tRNA expressing cells were able to recover the CFTR function close to the wild type level in black. So then we wanted to know if we can further improve ACE-tRNA rescued CFTR function by treating the cells with other pharmacological compounds like NMD inhibitor or CFTR modulators. So here So we delivered our ACE-tRNAs using adenovirus to 16 HB cells and treated with the SMG1 inhibitor or NMD inhibitor, and elexacaftor, tazecaftor, ivacaftor, or ETI as a CFTR modulators. And for when we measure the CFTR mRNA expression level on the left, leucine ACE-tRNA alone seems to significantly increase the CFTR mRNA expression level, and we were able to boost that expression by treating the cells with SMG1 inhibitor in gray bar. And ETI treatment didn't have any effect on mRNA level. But then when we performed the functional studies using Usin chamber, we were able to get this result. So the representative traces on the bottom and the total area under the curve quantification on the right. Again, leucine HTRNAs were able to significantly improve the CFTR function, but SMG1 inhibitor treatment had no effect. However, ETI treatment significantly improved the functional level of the CFTR. So from this experiment, overall, we got the best recovery of the CFTR function with the combination of all three, but the effect of SMG1 inhibitor was minimal. So up until this point, we wanted to insert a specific amino acid for a specific mutation site. But we started to wonder if we can use one HTRNA sequence for multiple CFTR nonsense mutations. So we tested this 4 most prevalent CFTR nonsense mutations with these 3 different HTRNAs, arginine, glycine, and leucine. The reason we choose arginine HTRNA is because we had very good arginine HTRNAs, and arginine to stop mutation is the most common nonsense mutation. And glycine HTRNAs because glycine is small amino acids, so it could work well in different positions. And leucine HTRNAs because leucine HTRNAs can suppress all 3 stop codons, and we have really good leucine HTRNAs in the lab. So we tested this idea first in the 16HB cells. So again, we delivered our HTRNAs using adenovirus, arginine, glycine, and leucine, and we measured the CFTR mRNA expression level. We performed the experiment in the absence and presence of ETI treatment. And as you can see, the CFTR mRNA recovery seems to be varied, but overall, with the leucine and arginine HTRNAs, we were able to achieve significant recovery of CFTR mRNA expression. And when we performed the functional studies using leucine chamber, again, leucine and arginine HTRNAs significantly improved the CFTR function, except for W1282X mutation, where the arginine delivery to the cell lines was not able to recover CFTR function unless we did the ETI treatment. Excuse me. So this suggests that W1282R needs some sort of assistance from CFTR modulators. Then we wanted to test our HTRNA technology in more clinically relevant setting. So in collaboration with the Big Man Lab at UMC Utrecht, we deliver our leucine HTRNAs to patient-derived intestinal organoids. So, okay. Again, we measure the CFTR mRNA expression level on the left, and we were able to rescue that level, although this level was lower than SMG1 inhibitor-treated alone condition. This result is still meaningful because we had only about 40% of transduction efficiency. And on the right, we performed the Forsgren-induced swelling assay to measure CFTR function. So we, again, delivered leucine HTRNAs to the organoids, and we separated the organoids into mTOR positive and negative populations. The positive representing HTRNA transduced, negative representing HTRNA untransduced organoid. So in the absence of Forsgren treatment, there was no functional channel. There was no functional detection. Yeah, but in the presence of Forsgren treatment, we were able to see HTRNAs significantly increase the functional CFTR level. We performed same experiment in different homozygote organoids, and for all different conditions, we were able to significantly rescue CFTR function with leucine HTRNAs. And when ETI treatment was added to the leucine HTRNA transduction, we were able to improve that functional recovery. Lastly, in collaboration with CFTR lab, we tested our HTRNAs in human primary enteric monolayers, or HPEMs, derived from intestinal organoids. which allow us to do 2D culture of the cells and perform functional assay. So here we deliver different dose of leucine ACE-tRNAs to G542X R1162X compound heterozygote HPEMs and measure the CFTR function. And as you can see, we got dose-dependent increase in the CFTR function with leucine ACE-tRNAs. And what's interesting is that if we use the lowest amount of MOI of leucine ACE-tRNAs, and when we add the elexacaftor and tezacaftor treatment, we were able to further improve that functional level, which was comparable to the higher MOI leucine ACE-tRNA transduced organoids, HPEMs, sorry. Yeah. We performed a similar experiment in G542X R553X compound heterozygotes with arginine and leucine ACE-tRNAs. And we got pretty similar result where we see dose-dependent increase in CFTR function and slight increase with ETI treatment on top of the ACE-tRNAs. So in conclusion, we were able to rescue the CFTR function by delivering specific ACE-tRNAs to specific mutations. But what I showed today shows that leucine ACE-tRNAs can successfully rescue the CFTR function for all different mutations. And this is really interesting because if we can, this makes ACE-tRNA technology more clinically available, as we only need like one FDA approval if this works instead of multiple different approval. So we hope that leucine ACE-tRNA works in other CFPTCs as well as other nonsense-associated diseases. So with that, I would like to thank our lab, the Big Man lab, especially Sasha, for performing all the organoid experiments, a CFFT lab, especially Kevin and Priyanka, for conducting and planning the HPEM experiments and funding sources. And I also would like to thank the organizer for giving me this opportunity to share our work and everyone who's here. Thank you.
Questions? You faked us out. Jay?
Thank you. Nice talk. Jay from CFTR lab. Interestingly, the ACE-tRNA is target the UGA, and as you know, the half of the transcripts has a UGA, but not UAA or UHG. So potentially there is a kind of off-target effect at the the normal stop codon if you use the ACE-tRNA, the UGA-tRNA. So is there any good idea to improve the specificity to PTC, CFTR PTC, PTCs? Yeah.
So you're asking if it's possible for ACE-tRNAs to target NTC over PTC and that could have negative effect? Is that your question?
And is there any good idea to target the CFTR PTC, for example, conjugated with the guide RNA? I'm not sure the kind of guide sequences to CFTR mRNA with the ACE-tRNA. Just for example.
Well, I've never thought about that way, but that's, yeah, certainly sounds like the possibility that we can explore. It seems like ACE-tRNA itself that really are more prone to interact with PTC than NTCs, and we could probably target it for CFTR PTCs instead of other like NTCs, but that's not something that we are currently exploring. As of this time, we are trying to make ACE-tRNA more available therapeutics and wanted to test in different cells, but that's certainly that we can do as we move forward. Yeah, I don't know if that.
So can I have one more question?
Yeah.
So have you ever tried any combination with the other the RT modulator with the StRNA?
Other modulators?
For example, G4NA or the ERF1 regulator, 3 regulator.
So we haven't really tried it with other modulators. We tested we tested the G418, but G418 itself seems to have negative effects. So we did not add another possibly non-native things to the cells. But E1F1
Release factor.
Release factor 1 degrader. That's something I think we are working on in collaboration with David Pebble group, I think. Yeah. So I think we would like to try with other PTC read-through drugs, but as of right now, ACRNA itself seems to work really well, and hopefully we don't need to use other factors to get good amount of read-through.
Right. Yeah, thank you.
Thank you for a great talk. So if your StRNA does target canonical stop codon, it will induce another mRNA decay pathway. It's called the non-stop mRNA decay. Did you have any readouts for that?
Not really. Not at the time. But
Just say no.
no. Sorry. Yeah, I'm trying to.
Thank you.
Any other questions? All right, let's thank Rory for another great talk.
Yes. Okay, our last speaker is Dr. Araf.
Nope.
No, Dr. Gillihat.
Yeah.
Yeah.
Yeah, sorry. Her topic is SPR 84 antisense oligonucleotide modulating the 3849 plus 10 kb CT splicing defect efficiently penetrates and restores CFTR function when treated through the apical side of primary HBE cells. Okay.
Thank you, and thank you for the committee for the opportunity to introduce our work. I would like to start and to dedicate my talk to my sisters and brothers, 1,500 babies, toddlers, young and old men and women that were raped, tortured, and massacred, and 240 young children, men and women that were kidnapped and are being held as hostages in Gaza by the Hamas, ISIS terrorists. I'll use this.
Maybe the first one you do. What the?
Let me do it this way.
Yeah, there you go.
Okay, so today I'll be talking about SPL84, antisense oligonucleotide for treatment of CF patients who carry the 3849 mutation. I'll be focusing on the preclinical pharmacological work that we have been working on for a few years now. It doesn't need to.
Yeah, that's a pain.
No, it doesn't.
Hold on.
Okay.
There you go.
Yeah.
Just like the balls.
Okay, so just to remind everyone what antisenses are, we're talking about short RNA sequences that specifically bind to the target RNA or pre-mRNA in the nucleus or in the cytosol and modulate the target RNA and the protein, leading either to decreased expression, to activation, restoration of activation, and overcoming mutations. Well, it doesn't work.
So don't use the.
Okay.
Yeah, just use this.
Okay, so Splicense is really a company that uses antisense oligos for treatment of pulmonary diseases. And we have a diverse pipeline. We are focused on specific unmet cystic fibrosis mutations, but also on large unmet still pulmonary diseases. Just to go back and talk about the 3849 mutation, we need to remember that we are talking about 1,500 patients globally that currently don't have a specific approved treatment. And when thinking about the current treatments in the US, the Kalydeco and the Symdeko are approved for those patients. However, it was demonstrated clinically that the effect of those two is very modest or disappointing. And this is why we're thinking about the benefit versus the cost. Those drugs were not approved in Europe. So what is this mutation? This mutation leads to an insertion of an abnormal exon or a crypt exon into the sequence of the CFTR mRNA, leading to a non-functional protein. Our antisense oligo specifically inhibits the insertion of this crypt exon, leading to fully functional wild-type CFTR. And this is really critical. You will see later on that this really generates a very potent antisense oligo combined with its long-acting properties, which has a great potential for those patients. But before I show you some pharmacological data, I would like to talk more conceptually about the delivery to the lungs. One of the main challenges of treating CF patients is really the fact that they generate hyper-concentrated mucus. And within our work, we really had to demonstrate that our ASOs can easily migrate, penetrate, and get to the target cells within the lungs, starting from the upper as well as to the still regions of those lungs. We did a lot of work, and I will share some of it, but before I do that, let's talk about what are the properties that actually generate a very promising ASO using our platform technology for delivery. The first thing is really our proprietary algorithms that really enable us to design and optimize our antisense oligos, not just for activity, but also for a better pulmonary delivery. For example, we're really focused on a very short sequences, ranging between 18 to 22 bases, which generate an antisense oligo at the size of 1 to 3 nanometers. If you look at the right-hand side, I think, you see how the mucus looks like. It's a pore-like structure. It is estimated that under obstruction, the size of those spores is around 50 nanometers. Our ASOs are much smaller. The second was to really optimize the chemical modifications that generate a stable, long-acting ASO, and this will be demonstrated as well. And lastly, we are using chemical modifications that generate a negatively charged ASO. So when thinking about the mucus, which is also negatively charged, you will see there are repulsion forces that further drive our ASO through the mucus. In this slide, you can actually see studies that were conducted in a muco-obstructive-like animal model, the beta-ENaC, which actually generate a lot of hyperconcentrated mucus. Those animals were treated with our ASOs and by intratracheal administration, and we then did a specific hybridization staining. As you can see, there is a very wide and robust distribution of our ASO within those animal lungs. When you zoom in, you can see that our ASO is actually detected within the different regions and sections of those beta-ENaC animals. It starts from the trachea down to the alveoli, and those epithelial cells are properly stained, which is comparable also to wild-type animals. In addition, we ran a study in which we evaluated the long-acting properties of our ASO. In this study, the ASO was treated only once, and 4 weeks later, we sacrificed those animals. As you can see, after 4 weeks, we still have a very strong and robust signal of our ASO within those regions, within those cells. We addressed this assay differently. We took a fluorescently labeled ASO, the SPL84, and we treated those animals again. But then upon staining or upon evaluation of the fluorescent staining, we also did a co-staining with the DAPI, with the nuclear stainer. As you can see, when we're looking at the merge of those two, a very clear and robust detection of our fluorescently labeled SPL84 on top of the nuclear stainer, suggesting that our ASO not just get to the epithelial cells, but also can be detected within the nuclear, where it needs to modulate the RNA. Lastly, when thinking about distribution, we ran a study in non-human primates. Those animals actually inhaled our ASO once a week for 4 weeks. And again, we sacrificed the animal and detected by staining, hybridization, our ASO within those animals. And you can see a very robust staining starting from the proximal down to the distal region of the lungs. You see a very strong uptake of our ASO in the bronchi down to the alveoli. And what's nice about those studies is that we were able to sub-characterize the epithelial cells that actually uptake our ASOs. You can see in black the goblet cells, for example. You can see the staining within the ciliated cells. We also have data for secretory cells. So we do have a very robust detection, penetration, and migration of our ASO, SPL84, within the relevant animal models. So what about the function? We did a lot of studies in which we evaluated the activity of our ASO using the gold standard Using assay. It was described before. What is the methodology? We use the same methodology. But here you see data in a huge amount, I would say, of different patient-derived cells, starting in HBs of homozygote CF patient, going into HNEs in heterozygote patients, and also in HBEs in heterozygote patients. I would like to point out that when you look on a homozygote patient, you can actually see that we have full restoration, almost 100% of the CFTR activity, as opposed to the Symdeco that had no activity within these patient-derived cells. And again, when you look at heterozygote patients, we did observe a very nice, almost full restoration, which is around 50% of the CFTR activity. However, this treatment was given by free uptake, and it's important, through the basal, using a basal approach. And when thinking about the cells and thinking about the treatment regimen in patients, we should address the apical, right? So we did those studies. We continue to evaluate the capability of our ASO to penetrate through the apical sides, to penetrate to hyper-concentrated mucus by free uptake, no transfections, to cells. In this study, we used an fluorescently labeled SPL84. It was actually given on top of the mucus. Those patient-derived cells secrete a lot of mucus, and the fully differentiated cells that secrete this mucus were treated with our ASO, and then we evaluated the staining of the nucleus within those cells. As you can see, there is a very efficient staining of our ASO within the nucleus of those HB cells derived from patients, suggesting that our ASO not just migrate freely and effectively through mucus, but also gets to the cells and gets to the nucleus. And this is really a durable effect. You can see that even when we follow those cells for 3 weeks post-treatment, still we were able to detect a very nice and robust signal of our ASO within those cells, within the nucleus of those cells. And it was translated to pharmacological effect. When you treat those cells through the apical sides, you see, again, the effect and the inhibition of the insertion of this, I would say, abnormal sequence or abnormal exon, because you have a correctly spliced CFTR as observed by the RNA, which is translated to a full restoration of the CFTR activity, which is comparable between the basal treatment to the apical treatment. So those studies were further conducted and actually expanded by the Cystic Fibrosis Foundation, by the lab of Martin, and I would like to thank the team. They took our ASO and further characterized the activity of our ASO when treating HBE cells, either heterozygote or homozygote, and further comparing the apical treatment to the basal treatment with our cells, with our ASOs on those cells. Again, they were able to detect a very robust effect and CFTR activity when treating with our SPL84, whether it was given by apical or basal treatment on both different HBEs. And another interesting thing that I would like to point out is when they did a head-to-head comparison of our antisense oligo to Trikafta on patient-derived cells that on one allele carries the F508del and on the other the 3849, you can see that our ASO was superior significantly superior in its capability to restore the CFTR activity compared to Trikafta, both when treated by apical approach or by basal approach. And in addition to that, and I don't show this data, the Trikafta had no effect on 3849 homozygote HBE cells as expected. So this is really about the capability to treat by free uptake, to migrate through mucus. But I would like, in my last few minutes, to really talk more conceptually about different approaches. We heard before about LNPs, and I wanted to share with you a study that was done in collaboration with Professor David Hill from the Lung Institute at UNC. He actually tried to compare the migration capability of our ASO to LNPs. Standard LNPs, which are commonly used, mainly consisting PEG and other lipids, and an average size of around 50 nanometers. And in those studies, he evaluated the diffusion coefficient, as you can see here. And he did that in CF patient-derived mucus, which consisted around 4.6% of solids. It's a moderate obstructive-like mucus. As you can see here, the migration or the diffusion coefficient of our ASO was 100 times better or faster through mucus. And when you look at hyper-concentrated mucus, which consist 8% of solids, the LNPs, which were fluorescently labeled, were practically stuck at the top of the mucus and were unable to migrate through the mucus, suggesting it's a huge challenge and barrier as opposed to our ASO that migrate very smoothly and fast through the mucus. When he did the calculation, okay, so how much time when thinking and knowing about the lung structure, the ASL, and actually the thickness of this layer will take our ASO to migrate through moderately concentrated mucus, they calculated and concluded it will take less than a minute as opposed to LNPs that will take them over 3 hours. And it's a big challenge when thinking about patients that frequently cough, trying to get out this mucus and to have a better breathing capabilities. So just to summarize, and I apologize for the format, something went wrong. We were able to confirm that our SPL84 is super potent and can really fully restore the CFTR activity in the gold standard Using assays, has a very robust distribution capabilities and migration. The safety profile, which will be presented tomorrow, was shown to be very promising in the toxicological studies. Phase 1 was successfully completed. There is a poster that actually shared this data. And we are now heading to initiate the phase 2 study in 3849 CF patients globally. And this study will be starting next year. We are aiming to treat those patients once a week by inhalation. The inhalation takes less than 8 minutes. And I do believe it's a promising approach. Thank you very much.
Questions? Gary?
Very nice. The issue, though, with 3849 is it's a leaky splice site mutation, and the efficiency of splicing varies amongst humans, right? So you showed a case where the homozygote didn't respond to the correctors, but we also know other cases of that similar genotype or same genotype where some transcript is made, wild-type protein is made, and that wild-type protein responds to the Trikafta.
So I'm not aware of Trikafta. I mean, obviously, we've been testing that for few. It's not just an example. It's really...
But my point is this, and I understand, I'm just thinking through this with you. Are you going to actually look at the efficiency of splicing patient by patient so you understand whether or not you're trying to correct someone with very inefficient splicing at 3849, or are you going to try to, characterize it somehow? Because the lesion is not uniform in its effect in different individuals.
So the answer practically in clinical studies, no, we're not going to do that. We cannot do that practically. We are looking on function, on effect on, FEV1, but I don't think we can actually look at that. It means that we need to do bronchioscopies, collect cells, and it's challenging.
Well, you get nasal epithelial cells. They could test a level of splicing, right? My reasoning is the following, is that I'm convinced this works. It makes a lot of sense to us. The question is, if you only have modest response in some people, that may be because they've already got quite a bit of efficient splicing and they're making quite a bit of wild type. The people who might be benefiting the most. So those are the inefficient splicers. And now you're recovering maybe from 5% to now you're getting them 20, 30, 40% of wild type. So again, I'm thinking it may help us characterize the patients ahead of time, even patient by patient, to know what kind of recovery we might expect with these kinds of drugs.
Thank you.
Yeah, I think that was I was going to follow up. So just trial design and how you're picking your patients, I think it could lead to success or not success based on splicing efficiency baseline between each patient. So you're not doing any, you're just basing it off function, not off of splicing.
Yeah, we're monitoring the function as inclusion criteria.
Yeah.
Hi, thank you. That was a wonderful presentation. I was wondering if you could share how you decide the timing to redose. Because when we think about treating our patients, if you deliver something into the airway and you just leave it there, patients are having airway clearance therapy 2 to 4 times a day. So if you multiply that by 7 days, it would have to be an extremely effective means of transfection for it to, within that very small interval between therapy, to actually be delivered to the right site. So I was wondering if you could share a little bit about how you decide that.
So we have done some work, obviously theoretical, because there is no animal model that can help us do that, the allometric conversion as we usually do, to try to evaluate it. It really goes back to the kinetics that I showed you some of, thinking about how long and how frequently and how much time we need it is detected. It goes to migration through mucus. We've done additional studies in CF patients. It's very fast, very effective, and it's distributed quite nicely. So we do believe that weekly will be enough to support that and to get this maximal exposure that you would like to see in your patients.
Okay, why not sooner?
So I think kind of optimizing. Obviously, we are treating with ASOs. We want to have on one hand, a very robust safety profile. On the other hand, have a very good and maximized efficacy. So we think that based on the data that we have collected so far, weekly will be good. And I can even say that we have data for other programs that we can go frequently.
Follow-up question to that? I'm right next to you. If you thought about front loading, so if you, or the benefit of front loading, where you would have maybe delivery every couple of days for then mucus for removal, and having more sparse delivery later on.
Yeah, I know. It was done before from doctors. We don't think it's needed. That's your study, which is longer.
Yeah.
Like more 8 weeks. So I see that will take time, but I think we'll get.
Any other questions? All right, let's thank the speaker for a great talk.